Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. I am Diaste Kananda Anindya-Swari. Would like to present our research with the title of Genetic Diversity based on RAPD F2 generation of apple cactus or Cereus spp. First, here's the background of this research. One kind of ornamental cactus, which is Cereus, is known and widely planted in Indonesia. Physical appearance of Cereus look like a tree that has a grayish-green cylindrical spines in diameter of 10 to 20 centimeter, more or less 10 meters in height, produce fruit with fruit skin colors that vary from purple-red to yellow. There are two types of apple cactus grown in Indonesia, which is short spines and long spines. For further information, apple cactus are self-incompatible plant, or means that unable to be fertilized by its own pollen. Apple cactus currently has a high economic value, but research on its genetic diversity has not been done much. Analysis of genetic diversity needs to be done to obtain specific characters of each genotype, which is morphologically and molecularly. Molecular marker can overcome these shortcomings by carrying out DNA-based analysis. DNA markers are more consistent and have high accuracy. PCR RAPD is one of the tools in molecular analysis by using certain markers in studying genetic diversity. It used to identify genotypes based on differences on DNA bands that have been amplified by random primer. The aims of this study is to examine the genetic diversity of the generation of apple cactus using RAPD markers. Next, for the methods of this research was carried out at the Laboratory of Genetics and Plant Breeding, Faculty of Agriculture, Gadjah Mada University in October until November 2020. The material used were 15 assays from the cross of the apple cactus, 5 samples each from the cross of long and short spines or GXP, the reciprocal or PXG, and long spines natural cross or GOP, selected from the greenhouse of the Faculty of Agriculture, 11 Maret University, Surakarta. The RAPD primer used were OPN5, OPM10, and OPD11. Materials were prepared for analysis of molecular characters following the DNA isolation protocol Doyle and Doyle 1990, added modification with the addition of polyvinyl pyrrolidone or PVP. The result of DNA isolation were carried out by PCR RAPD reaction with PCR composition per reaction using 12.5 microliter consisting of kotak green PCR master mix, primer, NFW, and DNA sample. PCR amplification was performed using a Biorad T100 thermal cycler observed by electrophoresis on 1.5% agarose gel. Electrophoresis is regulated with a voltage of 100 volt, 400 milliampere for 60 minutes. Fluorescence staining was applied so that the DNA band patterns could be seen under the UV light. Data analysis was carried out by scoring for the emerging bands. Furthermore, the scoring data were analyzed using anti-SPC software version 2.02 to determine genetic similarity or dendrogram, based on simple matching coefficient with the calculation of SIMQual. Then the cluster analysis was made by calculating the SAHN based on the UPGMA of the similarity index. Next, about the result and discussion of this research. First is about concentration and purity. Modification on isolation protocol of Doyle and Doyle 1990 by addition of polyvinyl pyrrolidone in purpose to reduce phenolic. This modification affects the result of purity ratio of DNA among 1.5 to 2.2. DNA purity belonging to good category if it ranged from 1.8 to 2.0. If the DNA purity value is less than 1.8, it means that contaminants in the form of protein are still found, while a DNA purity value is more than 2.0 means that contaminants are still found in the form of phenol. Then for PCR amplification, as shown on figure 1, showed that electrophoresis were carried out after successful DNA amplification. The result of band pattern has variosity. Therefore, the scoring is done on a band pattern that has strong intensity and brightness. Based on the visualization of DNA bands by the 3 primers, it can be seen that the intensity of the light produced is different. Some bands are faint and some are bright. The intensity difference in the resulting band can be influenced by the distribution of primer attachment, concentration, and genome purity. Polymorphism analysis as shown on table 2, the total number of bands generated from 3 primer use is 28 bands with size ranging from 300 base pair to 1,800 base pairs. The 100% polymorphism percentage belonged to the OPN5 and OPD11 primers, while the 83% polymorphism percentage was found in the OPM10 primers. The result of a high percentage of polymorphism indicated that the 3 primers were suitable to be used as markers in the genetic diversity of apple cactus. Next, for genetic diversity based on RAPD markers, as shown on table 3 and figure 2, the dendrogram with the simple matching coefficient resulted in the similarity coefficient ranging from 0.61 to 0.89. The clustering analysis resulted in 4 clusters. The 5 samples of short and long spines crosses, or PXG, form a group because they had similarity coefficient of 0.69. The second and third sample have the highest coefficient similarity among all cross assessment, which is 0.89. This grouping can occur because of the similarity of morphological characters. Morphological characters caused by the samples that grow close together or come from the same parents can trigger grouping of the samples, both from the same or different cross groups. And for the conclusion, RAPD can be used to classify apple cactus based on their genome. All samples of short and long spines crosses, or PXG, were clustered in the same clusters. The dendrogram with the simple matching coefficient resulted in the similarity coefficient of 3 primers ranging from 0.61 to 0.89. The more primer used, the more visible the diversity display because the more genomic segments read by the primers. Here's the reference used for this presentation. That's all for my presentation. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.